proteins perform their task by interacting with other molecules. Today I am going to talk about protein-ligand interactions. So, today I am going to talk about these few concepts. I will discuss what is binding affinity, what is specificity. I will give you examples of protein small molecule interactions, and I will also give you examples of protein-protein interactions. And in the next lecture, I will talk about protein nucleic acid interactions. So, these are some examples of protein ligand interactions. One example is where enzymes are natural catalysts. So, most enzymes are proteins and they facilitate or they speed up various biochemical reactions that occur. In order to do that, they have to bind the substrate. So, the first step of any enzymatic reaction is the formation of this enzyme substrate complex. So, this is one example of protein ligand interaction. I will discuss enzymes in great details in next week's lectures. When some enzymes do not work or do not perform their task properly, then it is required to inhibit those enzymes. So, drug molecules or most of the medicines that we take are some form of inhibitors. In this case, these are not substrates, but they look very similar to the substrate. So, in this case, these small molecules, they also bind enzymes and they inhibit the function of the enzymes. So, this is also another example of protein ligand interaction, and we will see one such example today. Apart from that, proteins can also bind to DNA. This is a double stranded DNA and this is a protein. So, this protein called a transcription factor, it binds to DNA in very specific sequence and then it will regulate the activation or the repression of downstream genes. So, transcription factors regulate gene expression and that is done by their specific interaction with specific DNA sequences. Then we will also see later on in this course that proteins can interact with other proteins or other small molecules. So, this Y-shaped protein that you see, these are called antibodies and they can interact with other proteins or other molecules which are called antigens. So, antibody antigen interaction is another example of a protein ligand interaction. Another very important interaction is the interaction of proteins which are embedded on the cell membrane like this. So, these are cell membrane proteins and these proteins have a special name called receptors. So, these proteins bind to other small proteins which can be hormones, and once they bind, there are some changes that are transmitted through this region down here and they get activated. So, we will see examples of protein tyrosine kinase where they get activated and they will phosphorylate their substrate which results in downstream signaling. So, the hormones, they act as signaling molecules, they will bind to their receptors and that will result in certain signaling events inside the cell. So, this interaction also an important protein ligand interaction, and we are going to see one example of this type of interaction today. So, let us look at the general features of protein ligand interaction. One type of protein ligand interaction is this, where the protein is there and it will bind to the ligand and it will form the protein ligand complex. So, typically we represent it by this equation where P is the protein, L is the ligand. So, both are free and then they come together to form this protein ligand complex. There is a second type of interaction which is called allosteric interaction. In this case, we may have two ligands and they have two different binding sites. So, ligand A binds to this site of the protein and this, once it binds to this site of the protein, it results in a change in the other site of the protein. So, this is called allosteric communication. So, you can see that the shape of this changes. Now, it can bind to the second ligand which is called the ligand B. So, then we have this complex where the protein is bound to both the ligand A and the ligand B. So, we can write it in this manner: there is protein, there is ligand A, there is ligand B. Ligand A binds to the protein first, results in some conformational change, and then only ligand B binds. So, this is called allosteric interaction because binding of the first ligand results in a change in the second binding site which is at a distant location. So, this is called allosteric communication and now the second ligand binds. So, I will talk about examples of this allostatic interaction in the next lecture in more details. So, today we are mostly going to focus on non-allosteric interactions. And if I talk about non-allosteric interactions, it can also be classified into two types. The first type is called lock and key binding. So, in this case the protein has a particular shape where the ligand binds. So, we can call this as the active site. The ligand has a particular shape, and you can see that these two shapes are complementary of each other, and then the ligand binds and it fits perfectly into this active site. So, this type of interaction is reminiscent of a lock and key interaction. So, there is a key which has a perfect complementary shape to the lock, so that the key will bind the lock. Another type of interaction is called induced fit binding. So, in this case the active site is slightly different than the shape of the ligand. So, you can see that this is not complementary to this. So, when the ligand comes, it will most probably form some loose interaction, and that results in a conformational change of this active site, so that it will look very similar to this, and then the ligand will fit. So, this binding induces a change in the active site, which results in perfect fitting or perfect binding of the ligand. So, this type of interaction is called induced fit binding, and examples of both are seen throughout different interactions in nature. So, let us talk about the thermodynamics of this interaction. So, here we have two entities, the free protein, the free ligand. So, let us say the free protein has a free energy which is somewhere here. So, in the y axis we are plotting the Gibbs free energy, and the x axis are all these different conformations. So, this is the free protein, it has a certain free energy. This is the free ligand, it has a certain free energy, and if we add these two, we get the total free energy or the combined free energy of the free protein and the free ligand. Now, when this ligand binds to this protein, the complex is formed. If the free energy of this complex is less than this combined free energy, then we will call this as a spontaneous reaction, which means that when the ligand binds to the protein, the total free energy is decreased. So, this will be a spontaneous reaction, right. So, let us look at it in more details, and how can we quantify something like this. So, this is the free protein, this is the free ligand, and it forms the complex. So, there is something called dissociation constant that we use to define how tightly the ligand binds to this protein. And we define the dissociation constant by this, where P in this third bracket is the concentration of the free protein, L is the concentration of the free ligand, and PL is the concentration of the protein ligand complex. So, in the numerator we have the concentration of the free protein and the free ligand, and in the denominator we have the concentration of the protein ligand complex. So, you can see that this will have Kd will have the unit of concentration, because we have concentration multiplied by concentration divided by concentration. So, Kd will end up with a unit of concentration, and this Kd is related to the free energy that we saw in the previous slide by this equation. So, delta G is the free energy, it equals to RT, R is the gas constant, T is the temperature in Kelvin, ln Kd. Now, typically this Kd is converted to a dimensionless quantity for this equation, and we can see that if Kd equals 1, which means that this and this, so the numerator and the denominator are equal, then ln Kd or ln 1 is 0, so delta G is 0. So, if I bring up this diagram again, it means that this delta G binding will be 0. So, the energy of the combined protein and ligand in the free state versus the complex will be equal. So, this is 0. So, this is not favorable, but for this to be favorable, delta G has to be negative. So, if delta G is negative, then this is negative, which means that Kd should be less than 1, and I will show you typical values of Kd that are found in nature for different types of interactions. So, this is the thermodynamics. What about the kinetics? Because the ligand will bind to the protein to form the complex. So, that is the forward reaction, but then this ligand can also diffuse out because there is no covalent interaction here. So, whatever bonds are there. So, again these are typically hydrogen bonds, electrostatic interactions and hydrophobic interactions. So, it can also break those bonds and come out. So, that will be the reverse reaction, and that is the dissociation. So, the forward reaction is the association, the reverse reaction is the dissociation, and these two reactions are given by two reaction rates. The forward will be given by this reaction rate constant called K on, and the reverse one will be given by the rate constant called K off, denoted as K off. And we can write this kinetic equation where dPl dt is the rate of formation of this complex. So, the rate of formation of this complex depends on the forward reaction. So, the rate constant multiplied by the concentration of the free protein and the free ligand minus the reverse reaction. So, K off multiplied by the concentration of the protein ligand complex. At equilibrium, what we are going to get is the rate of formation of the complex and the rate of dissociation of the complex will be equal. So, this left hand side will be equal to 0, and what we will get? We will get K off by K on will be equal to PL divided by, so P times L divided by PL, right, or in other words that is KD. So, KD is related to this on and off rate constants as follows: KD equals to K off by K on. But remember that this is valid only under equilibrium condition. So, how do we measure this KD? I will discuss this in more details in the last week's lecture, where I will talk about different biophysical and biochemical experiments, but this is a brief introduction of all such experiments. So, typically we define or we determine KD experimentally using some sort of titration experiment, where we will have the protein and we will slowly titrate in the ligand, and we will measure the formation of the protein ligand complex using some means. It can be fluorescence, it can be, if it is NMR experiment, some chemical shift changes, if it is ITC, then change in heat. So, there are all sorts of experiments that we can do to estimate the formation of this protein ligand complex. So, typically what we have is, we know the concentration of the total protein, we do not know the concentration of the free protein or the bound protein, we know the total protein, we know the total ligand, and we can measure the concentration of the protein ligand complex, right. Now, if I go to my equation of KD, I do not know the concentration of the free protein or the free ligand, but I know the concentration of my total protein, which is the summation of the free protein and the protein ligand complex. This I know. I know the total ligand, which is the summation of the free ligand and the protein ligand complex. So, what I can do is, I can replace this free protein by this. So, free protein will be total protein minus protein ligand complex. Similarly, free ligand will be total ligand minus protein ligand complex, and you can see an equation like this. So, we know P total, we know L total, we know PL, but you will see that this PL, when we multiply this, it will form a quadratic equation, right. So, we can solve that quadratic equation and we will get something like this. So, from the experiment, from the titration experiment, we will estimate KD, which is unknown. So, that is sort of the basis for all the experiments that we do, and I will discuss specific examples in the last week's lecture. So, what are the typical dissociation constants or KD values? So, we can divide them into these 4 categories. So, KD again has the unit of concentration. Lower KD value means tighter binding. So, if you again remember that equation, delta G equals to RT ln KD. So, lower the KD value, more negative will be delta G, okay, which means that the complex will be more favorable, right. So, these are the following ranges. When the KD value is less than 10 to the power minus 8 molar, right, or less than 10 nanomolar, that is something that we define as very tight binding, okay. And in that case, that binding becomes sensitive to picomolar concentration of the ligand. So, even if the protein is ligand is in picomolar concentration, the protein can sense its presence, because it will bind some of those of that ligand. When the binding affinity is between 10 nanomolar or to 1 micromolar, so 10 to the minus 8 to 10 to the minus 6, that is what we called tight binding. So, the sensitivity is in the nanomolar range. 1 micromolar to 100 micromolar is moderate, and if it is greater than 100 micromolar, it is very weak binding. So, typically in biological systems, we will see binding affinities in the range of these three: very tight, tight, or moderate binding. And the experiments that typical experiment that I just explained in the previous slide, there to determine the protein ligand KD, the experiment is designed in such a way that the protein ligand complex is detected in this particular range. So, 1/10 of the KD value to 100-fold of the KD value. So, again I will discuss this in more details in the last week's lecture. So, here are some typical values. Enzyme substrate. So, this is enzyme ATP, but you can think of it as enzyme substrate. So, enzyme substrate interactions are typically in the millimolar to micromolar range, and the KD values will be in this range. Signaling protein, we will see one example of a signaling protein today. They are in the micromolar, or they can also be quite tighter than that. So, they will be in the micromolar range. Protein DNA interaction, transcription factors, I will talk about that in the next lecture. They are typically in the nanomolar range, and inhibitors that we design for molecules, so drug molecules that are used as medicines, they are typically in the nanomolar to picomolar range. One of the most tight binding interactions that is known in nature is the interaction of biotin. It is a small molecule that binds to this protein called avidin, and that is in femtomolar range. So, 10 to minus 15. So, this is one of the strongest known non-covalent interactions till date. So, let us look at an example of a protein ligand interaction. For that, we will look at this particular protein. So, this is again an enzyme. I will talk about enzymes in the next lecture. So, enzymes catalyze chemical reactions under very mild biological conditions. So, this is an enzyme, it is called a kinase. So, it is an ABL kinase, and what it does, it binds to ATP and it will phosphorylate other proteins which have tyrosine residues. So, what it does, so this is the side chain of a tyrosine, you see this OH group. So, this kinase enzymes like this, they will add a phosphate group like this on this OH group. So, this is called phosphorylation, and this is a very important signaling event. So, once these substrate proteins are phosphorylated, they can go and bind to other things, and they can trigger cascades of reactions which signal something inside the cell. The reverse reaction is also true. There are enzymes called phosphatase which will remove this phosphate group called dephosphorylation. So, but now we are going to focus on ABL, which is a kinase. So, it puts phosphate group on tyrosine side chains, and this results in certain functions. So, this kinase is involved in cell division and cell differentiation. So, division means one cell divides into two, okay, and differentiation means that in our body there are, so if you think about the human body, there are almost 200 different types of cells. So, there are stem cells, and they can differentiate into other cell types. So, that is differentiation. So, I will talk about that in more details when we talk about cell division in later weeks. Now, when this process works fine, everything is okay, but in some cases this process does not work properly, and that can result in various forms of diseases. One primary disease is cancer, where the cell division and the cell differentiation is not working properly. So, when ABL kinase, its function is hampered, it results in the development and progression of a particular type of cancer called chronic myelogenous leukemia. So, this is basically a blood cancer. So, in this case, the kinase is always on. So, again these proteins are not always on, they get turned on under certain conditions, right. But if they are always on, it means that the cell division will keep on going without any check and balance, right, and that leads to the formation of this cancer. So, in this case, it results in overproduction of myeloid cells, which are white blood cells in the bone marrow and the peripheral blood. So, which is the characteristic of this leukemia called CML. So, in this case, what we want to do is we would want to inhibit the function of this enzyme. So, we do not want to work it continuously, we want to regulate or we want to stop its function. We want it to stop doing this, right. So, how do we do that? So, for that we have to develop some small molecule inhibitors which will bind here. So, it will stop the ATP from binding there and it will stop this reaction because this phosphate group comes from the ATP. So, here again the structure of the kinase bound to ATP is shown, and again you will see that. So, we have discussed this in the previous lecture. So, you can identify alpha helices and beta strands. So, this is the cartoon structure of the protein, and this is the structure of the protein bound to a small molecule which is an inhibitor. So, this inhibitor is called imatinib, okay, and you can see that it binds to the exact same position where the ATP binds. So, if this molecule is bound there, it will not allow the ATP to bind, and typically the binding of ATP to this enzyme is in the millimolar to micromolar range, right. So, something in the range of, let us say, 10 to the power minus 5. Imitanib we will see binds with 10 nanomolar affinity. So, that is 10 to the power minus 8. So, this is at least 1000-fold tighter than this, which means that if we add a small amount of this inhibitor, it can completely shut down this protein and not allow binding of ATP. So, how does imitanib look? It looks like this, and if you look at this and if you consider the structure of ATP, you will see they look very different. So, how did people come up with a structure like this? So, that is something called drug design, and we will see it in this particular example. So, here are some of the interactions, and this is true for most protein small molecule interactions. So, this is ATP. So, you have the sugar, that is the A group adenine and the phosphate group. So, there are 3 phosphate groups, right, and there are some of these interactions which are shown. So, this is a hydrogen bonding interaction. So, this is coming from the backbone of the protein. So, there is a hydrogen bond donor from the protein, acceptor from the ATP, donor from this amine group of the ATP and acceptor here. So, these 3 hydrogen bonds, there are hydrophobic interactions with these and these groups, there are polar interactions with these polar groups. So, all of this together allows the ATP to bind, but again these interactions are not very strong. So, it results in a, let us say, 100 micromolar binding affinity. In case of developing a drug molecule, a molecule like this was developed which mimics this interaction. So, you can see that the same hydrogen bonding interactions are there. So, these 2 hydrogen bonding interactions can be seen here. There is no hydrogen bonding interaction here, but there is a hydrophobic pocket. So, this molecule is put so that we can get a good hydrophobic interaction, right. So, a molecule like this will be called a lead molecule, because this is where we get some binding interaction, it binds in the right place, which is very important, and now we can improve this binding. And you can see there are these R groups, R1, R2, R3. So, we can change these R groups and look for better binding interaction. So, once we get this lead molecule, variations of molecules are made where this R1, R2 and R3 groups are changed. So, you can have a library where you have 100,000 molecules with all different moieties in these 3 positions, and you check the binding affinity. So, you determine the KD for those molecules using some high throughput screening, right. So, we will see an example. So, this is our lead molecule. One of the library molecules, let us say, looks like this, where R1 is this pyrimidine ring, pyridine ring, R2 is just the hydrogen and R3 is this moiety. And if you measure the binding affinity, it is 10 to the minus 5, right. So, 10 micromolar affinity. Other molecules that, let us say, show up from this library look like this. So, this is 10 micromolar, this was less than 10 micromolar, this is 10 micromolar, this is 1 micromolar, which is getting better. And finally, we have something which is 100 nanomolar, so 10 times tighter, and then we have imatinib, which is 10 nanomolar, right. So, you can see that all these groups will result in sort of hydrophobic interactions, the hydrogen bonding is there. So, all these interactions are there. So, you have to screen a library of around 100,000 molecules to get something like this. So, most drug development are done in this fashion. Okay. So now let's look at protein-protein interaction. So in case of protein-ligand interaction, we saw that these ligands or the small molecules, they bind to a particular pocket, right? But in case of protein-protein interaction, they do not bind to a small pocket. In case of protein-protein interaction, we have a large binding interface. So protein-protein interactions will look something like this, right? So in this case, we are seeing the interaction of trypsin and its inhibitor. So in this case, there are certain things. So some of the salient features are written here. They involve large binding interface. If you look at this bound state, this looks very similar to the interior of the protein, right? But this interface has a dual purpose. When the proteins are isolated, they will interact with water, and then when they are bound, they will not interact with water. So they will look similar to the core of the protein. So we do not expect a large number of hydrophobic residues like we have in the core of the protein, but still we will have some hydrophobic residues, because that gives to something called specificity, which we will see in the next few slides. We will also see molecules like this. So positively charged molecules and negatively charged molecules, which will give us nice electrostatic interactions. So all of this plus the complementary of the shape. You see, the shape is also complementary to each other. So all of this will lead to strong binding. And the last thing that gives a very favorable entropy is the displacement of these water molecules. So these water molecules are squeezed out. So they have limited entropy in this region because they will have limited mobility. But when these two come together, they are released to the bulk water and their entropy increases. So that also helps in driving this protein-protein interaction. So I have already discussed affinity, which is how tightly two molecules bind to each other, but there is also something called specificity, and we will learn specificity by this particular example. So FGF are fibroblast growth factors. So these are growth factors which control growth of new blood cell, wound healing, embryonic development, and many other events in our body. They bind to these proteins which are called receptors. So these are transmembrane proteins. So you can see that, so this is the cell membrane. There is a domain which is extracellular, so this is outside the cell, and there is a domain which is intracellular, so this is in the cytoplasm of the cell, and this is the transmembrane domain which goes through the membrane. So the outside one binds to this protein, and the inside one is again a kinase. So it's also again this is a tyrosine kinase, so it will phosphorylate tyrosine residues of its substrate proteins. So when this FGF hormone or this FGF is available, it will bind to this extracellular domain and results in this dimerization. Once they are dimerized, these two groups come together, they get activated because they are kinases, so they will phosphorylate each other and then they get activated. So in this state they are not activated, in this state they are activated. So the binding of the ligand activates this and then it will trigger certain downstream signaling events. Now it turns out that there are 18 different FGFs in humans and they are of very similar sequence and structure. So you can see some of the structures are shown here and you can see that they look very similar. They have very similar beta strands, right, and some of these alpha helical turns, right. So they look very similar. So there are 18 FGF and there are 7 receptors. So if we consider all of them, all possible combinations, 18 of these growth factors can bind to 7 of the receptors, resulting in 126 different types of interactions, right. Now, not all cells will express all the growth factors and all the receptors, right. So a typical example is shown here where a particular cell type, let's say, expresses only these 4 receptors and these 6 growth factors. So again in this case 4 times 6 we can have 24 different combinations, right. Now if all of them are interacting with each other equally, then we are going to have a mess. So there will be no specific signaling, right. So for that what is needed is that these receptors should bind to these growth factors in some degree of specificity, and we will see some examples. For example, if I pick up these 3, So, these solid lines mean that there is a strong interaction, right. So, if you see 3, there is not much interaction with 1, 3, there is a weak interaction with 2C, 3 has a very strong interaction with 2B, and 3 has no interaction with 3C. So, which means that FGF3 is specific to FGFR 2B. So, this is a specific interaction. On the other hand, if we look at FGF1, then you can see that it is interacting with 1C, 2C, 2B and 3C, right. So, it means that FGF1 is not very specific with respect to these 4 receptors. So, it is the combination of all these interactions where there are some very strong specific interactions, they will determine the overall fate of signaling that is transmitted inside the cell. So, whatever you see here, so you can pause the video and look at it carefully, you will see that there are some interactions which are absent and then there are some interactions which are very strong. So, this is also shown in the tabular form here. So, this is FGF1 and you can see that it binds to these 4 receptors with very similar affinity 10 to the minus 7, 10 to the minus 7, 10 to the minus 7, this is 10 to the minus 8. This last one is actually an artificial receptor that was designed. So, FGF1 is not very specific, but if you look at FGF5, there is not much detectable interaction here, not much detectable interaction here or here, but it binds to FGF2C. So, FGF5 is highly specific to the receptor 2C, right. Similarly, 3 is highly specific to 2B. It binds to 2C with some affinity and it does not bind to 1C and 3C, right. So, this is called specificity, that protein-protein interaction and even protein-ligand interaction. So, you can, for example, an enzyme can have all sorts of molecules which are protein together that look very similar, but it will bind to only its substrate, right. So, only one kind of sugar molecule, it will not bind to other sugar molecules, right. So, this type of interactions are called highly specific interaction and this is something that is very important. So, in case of protein ligand interaction, there are 2 important factors. The first one is affinity and the second one is specificity. So, again these are the books that you can follow for the topics that I have covered, and thank you.